I hope everyone here in Chicago and everyone online was able to connect and network a little bit during the break, get a little refreshment. I'm personally really thrilled to introduce Catherine Saenz, PhD, RD, who will be presenting on the interplay of nutrition and exercise with human genomics and gut microbial communities. Dr. Saenz joined Jacksonville University as an assistant professor for the 2018 fall semester after completing a postdoctorate fellowship with the Cleveland Indians in nutrition and sports science. She specializes in exercise, metabolism, and physiology. Her main areas of study include the mechanism and applications of ketogenic diets and bridging the gap between exercise science and sport nutrition. Her research interests extend into how diet manipulation and exercise may affect human genomics and gut microbial communities. She's keen on how to best translate this information to the applied setting for optimal health, wellness, and performance. Dr. Saenz is especially passionate about sharing how exercise and nutrition can support each of these areas with the classroom, athletics, community, and minority Spanish-speaking populations. So this is a great combination of a lot of what we do here at the ANA with the omics, with ketogenics, ketogenic diet, and with nutritional genomics. So I'm really excited. She's also one of our associate editors for the journal. Welcome, Catherine.
Thank you all so much for joining us this afternoon, and a big thank you to the American Nutrition Association for this invitation. I'm excited to walk us through how ketogenic diets, gene expression, and microbiome, it's a big topic, how these omics reflect some of the phenotypes that we're also observing. Before we jumpstart into anything, I also want to make sure that I identify that I don't have any disclosures for the presentation, but I do want to emphasize that this presentation is very much off of evidence-based research and education. But when it comes to individualization and personalization beyond that, well, this conference is really speaking to how much more we need to look at a bigger picture. So we're looking at this very much through the research lens, and then we can talk through how we translate this information to the practical setting. Our learning objectives for today include defining nutritional ketosis and ketoadaptation, getting us all on the same page, as well as identifying the phenotypes that we observe in this particular metabolic state. We're also going to discuss the evidence that we have available to us and how the microbiome is impacted with metabolic health and the relationship there. And then we're also going to discuss what's known and really what's not known about the ketogenic diet and the microbiome. As the title reflects, we also had some data available to discuss some of the additional omics, and so that's where we get to jump into the transcriptome as well. And so really where we get to take our journey today is these very consistent phenotypic adaptations that we observe through habitual carbohydrate restriction and these metabolic and systemic effects that we're seeing with ketogenic diets. At different levels, we're also able to observe these adaptations, or are we? That's what we're going to dive into. And we're specifically going to look into the systems of skeletal muscle transcriptome levels, so gene expression levels at that tissue level, and then also extend it beyond into other systems that we are the host to, into our microbiome. And really the crux of the talk is how we're able to see our external environmental factors like habitual diet impact so many different levels of our physiology, and what this reflection may look like in turn in some of these adaptations and phenotypes that we observe. And so to start us off on what is a ketogenic diet, there's so many different operational definitions of a ketogenic diet. Today we're going to be discussing a ketogenic diet as it relates to a high-fat, moderate protein, carbohydrate restricted diet. So I emphasize that this diet typically ranges around 65 to 75%, and the versions we're going to discuss today, which is really focused on metabolic health and energy metabolism. And I highlight that because there are ketogenic diets that are much more specific to other paradigms. So for example, an individual that may be following this for epilepsy control may have a different dietary prescription. So today we're looking at a high fat prescribed at around 65 to 75% of the diet, moderate protein around that 20 to 30%, so there is still a moderate protein inclusion, and then of course carbohydrate restriction at 5 to 10%. I also want to emphasize that I'm putting in the carbohydrate at a percentage because I am going to be discussing healthy, active, athletic populations, as well as those that are looking to improve metabolic health. And while carbohydrate restriction and ketogenic diets are often prescribed at a carbohydrate gram amount, that doesn't always include all populations. And so in the essence of personalized diet, when we're able to take it more at this percentage range, we can really adapt the diet to the individual and find nutritional ketosis per the individual. Now, often when we talk about ketogenic diets, people commonly discuss this diet in a high fat, but tends to be a very high animal protein diet. And while animal protein can absolutely fit into a well-formulated ketogenic diet, it's not the only thing that it's included. In fact, a correctly formulated ketogenic diet, or what we're identifying as a well-formulated ketogenic diet, is one that's still rich in the essential nutrients that the body requires. So it still has the vitamins and minerals that we're looking for. It still has those essential fatty acids. And it still contains, while smaller and reduced levels of carbohydrates, there are still carbohydrates present in the diet. In fact, they look a lot more like your non-starchy vegetables and different ranges of fibrous fruits. So we have a very rich and very diverse diet inclusion. And I think that's really important for the populations that we're going to be discussing, that often we discuss ketogenic diets in a very restrictive manner, but it's not necessarily restrictive, it's just different. Just a different approach to how we're going into our diet. And this diet is not necessarily emphasizing a particular animal protein source. There are those that follow ketogenic diets plant-based, those that follow it animal-based protein source. We're really just looking at those macronutrient ranges that supply the body with the energy needed and then allows us to enter what's called nutritional ketosis. And so a lot of operational definitions to get us on the same page. Nutritional ketosis for us today is going to be defined at anywhere circulating ketone levels or capillary ketone levels identified at 0.5 to about 3 to 5 millimoles per liter. Research is identifying that really that sweet spot seems to be at 1 millimoles per liter and higher, but really that 0.5 to 3 range is nutritional ketosis. And nutritional ketones are, let's take a step back, ketones are a natural byproduct of lipid metabolism in all of us. Once we are in a state of carbohydrate restriction, that natural rise now starts to become an alternative fuel source. And so the body, now increasing its lipid metabolism, also increases its ketone production, but very much in a physiologically absorbed way. Therefore, nutritional ketosis, we're able to use these ketones as alternative fuel sources in multiple different tissues. And we're also identifying that this rise in ketones impacts the body in a very systemic way. After we're following a carbohydrate-restricted diet for anywhere between 3 to 6 weeks, we start to enter a state of ketoadaptation. And ketoadaptation is this umbrella term that really defines these distinct physiological adaptations that include energy metabolism, they include shifts in our fuel utilization, but also seem to be accompanied with very systemic effects. I want to make a note that this is distinct from ketoacidosis, which is a very serious metabolic crisis for the body. Ketoacidosis, these ketone levels at 15 and much, much higher. So nutritional ketosis is a natural physiological effect that the body goes through when we're in carbohydrate restriction, but not necessarily calorie restriction or any sort of metabolic dysfunction. And when it comes to ketoadaptation, while it is a topic that we're discussing more nowadays, it's obviously a topic that's been studied for extended periods of time. Most of ketoadaptation has been studied in the context of clinical diseases or disorders, primarily in neurological diseases, more specifically epilepsy. But we are starting to identify that, again, the systemic effects of nutritional ketosis and ketoadaptation allows us to also look at this in very distinct physiological states of the body and illness-related diseases. Well, I guess all illness is a disease. But we start to observe it in situations where the body may be undergoing some sort of a disease or disorder. And so this extends from epilepsy into a wide range of clinical implications, such as PCOS. We're starting to see some research increase. We're starting to see research increase in metabolic health. Substantially, we're starting to see research increase in oncology-based work, and we're starting to see research increase in these areas also in other behavioral and other neurological aspects. And so what's so unique about ketogenic diets is that we start to see that the application is very widespread, that this is a very unique metabolic state that starts to impact several several areas of the body. Therefore, it's being studied in several contexts. However, the phenotypes exhibited seem to be very consistent. And so a large number of studies, in addition to whatever the focus may be, are also observing what we're identifying as this keto-adapted phenotype. And some of the crux of those adapted phenotypes include a fuel utilization shift, so a reliance on fat oxidation and fat metabolism, and a decreased reliance on carbohydrate metabolism. We're not removing carbohydrate metabolism by any capacity, we're just not relying on it to the extent that we would in a different dietary profile. We also very uniformly start to see improvements in body composition, where we see specifically decreases in body fat. Interestingly, not always a decrease in total body mass, but there does seem to be a consistent decrease in body fat and a maintenance, and in some studies, depending on their dietary prescription, even an augmentation or an increase in fat-free mass, specifically lean mass. And so these body composition improvements are also a very consistent finding with keto adaptation. The metabolic adaptations have been well documented, especially in the past several years, where we're observing improved insulin and glycemic control, improved blood pressure management, and very unique cholesterol profiles. And so why I say unique cholesterol profiles is because we do seem to see consistently a decrease in triglycerides, an increase in our HDL, but often, in about half the population, those that have been studied, we also see an increase in LDL. This is typically seen in a negative context, but as we're starting to identify, it's a different context. So essentially stating that what we typically prescribe with, or how we typically read individuals on a high-carb diet, well, this is a distinct metabolic state. So we're unsure if those same values also have a negative outcome, or if they're just representative of a keto-adapted phenotype. But whichever direction they are heading, which research is diving into that and finding some really interesting areas to explore, we can agree that there are some consistent metabolic adaptations that occur in this keto-adapted state. The last piece of it, which is the area that I find most interesting, are these systemic physiological changes that we observe with keto adaptation. And this is where I find the power of diet really being able to expand in so many of the areas and topics that we hear, not just at this conference, but that we're starting to explore in clinical populations and in athletic populations. These systemic changes impact well beyond energy metabolism, and it's been documented that keto adaptation, and specifically that rise in ketones, is impacting our inflammatory pathways, impacting our immune systems, impacting cell signaling, and impacting oxidative stress pathways and antioxidative stress pathways. And so this is just one snapshot of several systemic impacts that we're observing in this keto-adapted phenotype. And what's so interesting and so exciting is that if we can see this, then we want to understand at what level are these changes occurring. So how is essentially our breakfast, lunch, and dinner, how is it impacting us, to what extent, and how can we then use this to continue learning more, prescribing correctly, and personalizing this diet to individuals that can fully benefit from it. And so what we want to learn are what are these underlying mechanisms that lead to what's been identified as these pleiotropic impacts, or these extensive, very unique, but very systemic changes that we're seeing and observing in individuals that are following those ketogenic diets for several weeks to several months. And so we observe these phenotypes, we notice these systemic changes, and I'm not sure if anybody here knows anyone that's followed a ketogenic diet for extended periods of time. The responses are often unique. We see these metabolic changes, but also we see these additional areas that people report. While that's anecdotal evidence, how can we bring that into the lab, study it more, and identify what are these possible epigenetic modifications we're observing, and then how does this extend beyond us to our first layer in our gut health? And so this work has actually been looked at in a couple of different models. It's fairly recent, within the past decade, but the models that have been looked at have found, again, that increase in ketones, and specifically beta-hydroxybutyrate, one of the ketone that's increasing the most during a ketogenic diet that we can measure in circulation. these particular markers seem to be direct epigenetic modifiers. So that has been identified, and it's mostly been identified in vivo and in vitro models, primarily murine models. But what we're observing is that in these particular settings, we can see these in a variety of tissues. So we can see these global gene expression differences when the subject population is following this particular dietary approach. In these lab settings, these are highly controlled settings. So the fact that we can identify it through this dietary approach and through ketone metabolism specifically allows us to identify further mechanisms. What we haven't been able to look at as much is how this impacts in humans. Now, there have been some human research. There has been some human research done. Primarily, it's been done in clinical populations, and it has had a heavier emphasis on epilepsy and neurological diseases and disorders. So the question that we had was, what about metabolic health? And also the population that doesn't get as much attention, what about healthy, active populations? Especially athletic populations, we find that at highly elite athletic populations, the body is still under a large number of stress from training. How do we help modulate that training? How do we learn more about what's happening metabolically, and how, again, one of these environmental factors, external factors such as diet, is impacting their metabolism, and can we use it to learn more about sustaining and supporting not just performance, but athletic development. So we had an opportunity to study just this question in what's called the FASTER project, which was an incredible project that we did that stands for Fat Adapted Substrate Use in Trained Elite Runners. It's a very long name, but the FASTER project dealt with ultra endurance athletes, so it fit perfectly. And this FASTER project, the primary aim was actually to look at metabolic differences between high carbohydrate-based athletes and low carbohydrate-based athletes. But we had an opportunity to do the secondary aim and not just learn what's happening at this phenotypic level, but also potentially learn about these underlying mechanisms. And so what we did during this study is we took 20 highly competitive ultra-endurance athletes. So our operational of highly competitive, these athletes were competing at the national and international level. Several of them were representing the country in their respective sport. These athletes commonly raced at lengths that were 50-kilometer or more, so they were getting after it. These athletes were also very specialized in their training. And by that, I mean that not anybody runs 50 kilometers or more in a casual sense. And so training is very, very specific and very voluminous in order to be able to compete at the levels that they're competing. These athletes sort of weed themselves out because of how specific the training is, how specific this type of competition is, and the other factors that are involved with it. And so for our particular study, we were looking for athletes that did have similar training, similar competition, and then also were placing in similar rankings at that competition level. The only difference that we wanted to identify, or the only obvious difference that we wanted to identify between the two groups, was their habitual diet. And so these athletes had to be following their preferred dietary approach, so that's a very important aspect as well. This was not an intervention. These athletes just compete at their respective diets and are able to place in equal sitting with both. Athletes were either following a high carbohydrate diet or a low carbohydrate diet. And if you're able to see the table here, really what we're identifying is there was no statistical significant difference between any markers that may identify something that may impact their performance per se. In fact, we wanted them to have similar performance levels and really differentiate them by what they consumed. And so on a habitual sense, these particular diets, the high carbohydrate athletes were following a very traditional high carbohydrate diet for an endurance athlete. Typically, that's prescribed around that 55 to 65%. These athletes were right in that range at 56%. They followed that moderate fat diet, which again is a typical prescription for an endurance athlete, with a moderate protein range at about 15%. Our low carbohydrate athletes were on the complete opposite side, and were really breaking the form of what's considered normal for a highly competitive endurance, especially in ultra endurance athletes. And so these athletes, as opposed to the prescribed 55 to 65% carbohydrate, completely dropped it down to 10%. So going back to those original slides on when we're doing percentages versus grams, this is a perfect example of when we can personalize those particular dietary ranges to the population we're working with. Now, grams are commonly used and the primary way to use, but sometimes we do step away from that. And these athletes are an example of that because several of them were actually consuming what we would consider outside of a nutritional ketosis range. However, due to their high level of exercise, that percentage still allowed them to maintain nutritional ketosis. From that, instead of that 29% dietary fat, these athletes were consuming on the complete opposite side at 71% fat. And then protein was set at 19%. And so there were no statistical significant differences between protein ranges. Really, the only difference that we found was between our two primary energy macronutrients of carbohydrates and fat. And what they did is a very fun protocol. It was a 2-day study for each subject. They came in and we flew them in from all over the country. They completed a VO2 max with us the night before so we could identify their 65%, right around that crossover effect, 65% VO2 max. And the next day was the experimental protocol for the results we're going to discuss. And so the experimental protocol really centered around a 3-hour run, or about 180 minutes. Pace was set at 65%, although athletes ended up running on average about 64% throughout the 3 hours. So they ran continuously at that pace. Now before, during, immediately after, and 2 hours after their run, there were several markers that were taken. And for our conversation, we were collecting oxidation rates throughout the study, and then we collected muscle biopsies immediately before the run, about an hour before the run, immediately after the run, and then 2 hours after. So 3 hours on the treadmill and a lot of poking and prodding, so a lot of fun. And what we found from this was that these particular athletes were exhibiting what we're again identifying as this keto-adapted phenotype of this increased fat oxidation and decreased reliance on carb oxidation at rest, throughout the 3-hour run, and into recovery as well. So throughout the day, they just were at an increased level of fat oxidation. They also were able to increase several markers, circulating markers that were based off of fat metabolism, and several circulating markers that were reflective of ketone metabolism. So we're starting to see this keto-adapted phenotype really come out. And we wanted to identify where else are we able to see some of these reflections? Are we able to see them at other levels of the tissue? And so from there, we were able to do skeletal muscle transcriptome analysis. We did this particular portion of the study at 3 time points. So again, at that muscle biopsy, about an hour before the run for a true baseline, immediately after the run, and 2 hours after the run, which is counting as our recovery period. We sequenced on the Illumina HiSeq 2500 and ran through our bioinformatics and our statistical approach. I just wanted to highlight that our functional annotations and our gene ontology focused on gene cards and David. So for the results, what we were able to observe, which was really exciting, is that at this level, even with how highly specialized these particular athletes are, again, we're trying to match them as much as possible to these external factors, we did see a divergent gene expression pattern when it came to the low carbohydrate versus high carbohydrate based athletes. We also observed these differences at each of the time points. So we could see that at the baseline time point, there were divergent gene expression patterns. Immediately after exercise, they upregulated distinct gene expression levels and distinct patterns. And then very interestingly, something occurred at that 2-hour mark as well. When we did some further analysis for the global gene expression, we found that many of the gene expression differences were primarily located in the mitochondria, and also the differences in the genes that we found were primarily related to energy metabolism. So this is on that global scale. When we're looking at the main effect differences, we found that 25,282 genes total were sequenced throughout the entire sequencing analysis. And of those 25,282, 653 were specifically designated to be impacted by diet, so about 2.5%. We also found that 8,400, slightly over 8,400 genes were impacted by the exercise, which really speaks to just how powerful of a stimulus exercise is. And then also we were able to identify even more that 312 genes were impacted by our diet by time interaction. So the next few slides, we're going to break it down by each time point. And what we're identifying baseline was really where we saw the most differentially expressed gene expression patterns, where we were able to identify the most annotations, and we were able to really go through that gene ontology in the most specific manner. And what we found is that when we set our standards, we got 33 genes were differentially expressed. And of those 33, over 50% of them were upregulated, specifically in the mitochondria in the low carbohydrate diet group. Of those genes, 35% were related to fatty acid metabolism. So we're seeing these gene expression levels related to fat oxidation, the same fat oxidation increases that we observed at baseline, we're also seeing them here in the skeletal muscle tissue. We also wanted to take note that over 80% of the genes that were upregulated in the low carbohydrate diet group compared to the high carbohydrate diet group were increased either 1 to 3-fold higher. So these weren't small differences between the groups, they were several-fold higher. When we jump into a comparison in how baseline compared to the immediately post time point, what we can observe is that 725 genes were increased 2-fold or more in the high carbohydrate diet group compared to 613 genes that were upregulated in the low carbohydrate diet group. So they had different genes that were upregulated, and also the function of these genes were very distinct. What we observed in that immediately post timeline is that the genes that were increased were primarily related with things having to do with exercise-induced muscle damage, inflammation, immune, and carbohydrate metabolism. Interestingly, though, the low carbohydrate diet group expressed genes that were more related to beta oxidation and fat metabolism and energy metabolism, as well as inflammation. What we're doing now in our analysis is trying to identify through some pattern-based software, where can we categorize these genes even more? And again, what's the functionality of the patterns that we're observing? So we can better understand what's happening in this state, how is diet influencing it, especially after such a strenuous stimulus of a 180-minute run. The most interesting finding was the 2-hour post. So this was counting as our recovery timeline. So 2 hours after the 3-hour run, and what we observed is that there's so much going on in the body, and now it seems that there is one distinct gene different between the two groups. And this particular gene, the PPP1R1A, is not only expressed differently at that timeline, but this particular time point, this particular gene was also expressed differently at each of the time points. And so if we're following the pattern, there was statistical significance at baseline in this particular gene. There was statistical significance immediately post, and then it's the only gene statistically significant 2 hours post. So not only is that really interesting when we dig a little bit deeper into our gene ontology to identify what this gene is related to, it's actually related to glycogen metabolism. And not just glycogen metabolism, but the hormonal control of glycogen metabolism. What's so unique about this particular aspect, there was one result that I didn't share. Well, there were several results I didn't share. The paper's fantastic. But there was one particular result that I didn't share for this study that's related to glycogen metabolism. We were able to assess skeletal muscle glycogen at each of the time points as well with that same muscle biopsy. And typically, one of the things that's stated about keto-adapted athletes is that while it may impact your fat oxidation rates, you also seem to decrease your skeletal muscle glycogen, and that may have an impact on performance and possibly recovery for subsequent bouts. We hypothesize what's been observed in the literature previously, that skeletal muscle glycogen was going to be statistically different between the two groups, and that the low carbohydrate diet group or the keto-adapted athletes would have a reduced glycogen metabolism or skeletal muscle glycogen level. However, that's not what we found at all. In our particular study, we observed that these athletes had similar levels of skeletal muscle glycogen at baseline. So despite their dietary differences, which is really where we put high carbohydrate diets in place for endurance athletes is to maintain that glycogen level. Now we've also identified several other reasons why we might follow that, but in particular that glycogen level, we're now seeing that these keto-adapted athletes in this particular study had no difference in their glycogen levels prior to the run. Then we head into our immediately post marker, and we find that actually after exercise, although they were relying so heavily on fat metabolism, they also decreased skeletal muscle glycogen to the exact same level, or no statistical significant differences between these two groups as their high carbohydrate counterpart. So we're seeing a similar pattern immediately post-exercise. And then the one that was particularly interesting is that we saw skeletal muscle glycogen replenish in the high carbohydrate diet group. That was expected, but we also saw it replenish in the low carbohydrate group despite not being fed any carbohydrates and despite relying heavily on fat oxidation. Data to support that was found through several oxidation markers and circulating markers. And so what was happening in glycogen metabolism is still something that we are we're not completely sure about and interested and excited to continue exploring that story. But something that was so unique in our finding was to follow that pattern, that unique pattern in glycogen metabolism, and then also have this particular gene be expressed differently at each of the time points. And so how does that connect? What does that mean? And how does that impact what we know about glycogen metabolism in the keto-adapted state? And also something to note is that this particular study looked at athletes that were already following their respective diets. And these athletes had to be on them for a minimum of 6 months, but the keto-adapted athletes on average were consuming their dietary approach for 20 months. So we're talking about close to 2 years with a range of 9 months to 36 months. And so these athletes were, again, self-selected, living in this particular state and competing in this particular state. So one of the things that this is starting to point to is that there are several adaptations that are occurring. We can observe these phenotypic observations, these phenotypic adaptations that we are seeing in other keto-adapted state, but also this extended period on a ketogenic diet. What are some of these other systemic effects that are occurring, and how are we shifting metabolism, and to what extent are we impacting our other systems as well? So our primary findings from this particular study was that we observed phenotypes consistent with the keto-adapted state. We saw this increase in fat oxidation and a decreased reliance on carbohydrate oxidation. We also saw genes that were reflective of these particular metabolic states. So we saw genes that were involved in energy metabolism and specifically fat oxidation rise and increase at all of these time points. And more specifically, we saw these particular genes increase at baseline, immediately post, and 2 hours post, all related either to beta oxidation or carbohydrate metabolism. And so a very, very specific metabolic state. And when we're looking through this information and how to properly contextualize it, really we need more studies to better identify this keto-adapted state and this phenotypes associated with it. But again, we can see these fuel utilization shifts, these metabolic adaptations, and these systemic physiological changes. And this was all done in the context of healthy, highly active, elite level athletes. But most of the work that we do is in clinical populations and trying to find ways that we can improve systemic health. And that's why we turn to diet, because it has that systemic impact. So we're going to shift gears a little bit and look at how keto adaptation may interact with the microbiome. And we're also looking at this in the context of metabolic health. And the reason why we're highlighting metabolic health is because ketogenic diets have been known to impact the microbiome. And a lot of this research is very specific to epilepsy. However, we haven't observed as many studies as it relates to metabolic health. And knowing the implications that we have in metabolic health and the implications that the microbiome has with metabolic health, looking into this further was a question that we really wanted to understand. And again, trying to understand, how does this keto-adapted phenotype, how does this keto-adapted state continue to impact so many different areas of the body? As we know, the microbiome is involved in everything from energy metabolism to immune health, even to behavior modification. So quite an active area of our bodies. And when we look a little bit deeper and we look through how diet impacts the microbiome, there's great appreciation for the fact that diet has a direct impact on the microbiome within just a couple of hours to a couple of days. But what's been found is that sustained impact on the microbiome is really coming from habitual diet. So not these short-term diets or short-term interactions, but it really has to be a chronic adaptation to a specific dietary prescription. And from this research, what's really fascinating is that we can identify what's considered as this healthy microbiome, as well as what's considered a dysbiosis, or when we start to see the differential patterns in either function or diversity in our microbiome. And this dysbiosis specifically is associated with chronic disease and autoimmune disorders and diseases. So I think the autoimmune part is newer research. We very closely related it to chronic diseases and disorders, but now also relating it just to how impactful the microbiome is to immune health and how that's extending into metabolic health. So when we're looking at metabolic syndrome, which is really our point where we're trying to pull individuals back and work through that preventative health, we find that gut dysbiosis is related to an increased gut permeability, a decrease in gut mucosal integrity, and also we start to see an increase in LPS in the gut and possibly into circulation as well. Now, this led us to study this particular question further and how the microbiome and how metabolic health may be interacting, or what's their relationship. And this particular study also published, the M.E.A.L study was looking at the metabolic effects of altering lipids. And the primary aim of the study was related to metabolic health and was related to macronutrient manipulation. So we wanted to learn more about how varying levels of carbohydrates and fat interacted with health. Now, a secondary aim was when we were able to look a little bit deeper, again, extending beyond the primary aims of the study into the microbiome. But because that wasn't the primary aim was really metabolic health, the study design reflected that as well. So a very unique study design. We were able to take individuals, 16 individuals in total, and completely control their diet in every single way during the duration of the study. Everything was controlled to 1/10 of a gram for that particular subject. So what we did is we took individuals with metabolic syndrome and had predetermined prescribed diets that started at a very low carbohydrate ketogenic diet for the first 3 weeks, which they completed on their own. And then for the following 18 weeks, they came to the lab and picked up food that were consumed in a controlled research kitchen. And so every single participant was consuming not just the same type of food, but also prepared the same way, and many times coming from the same grocery bag, if you will, essentially meaning that we weren't getting seasonal differences, we weren't getting differences in location, we weren't getting differences in their portions. They were really tied to the individual. So every individual was calorically controlled. So they were eukaloric diets throughout the 21 weeks. And of those, individuals were on a slightly hypocaloric for their particular diets at about 300 calories hypocaloric. And so these diets, which were highly controlled, highly measured, and very specific to the individual, the only difference was that while they started at a low carbohydrate diet, every 3 weeks or about every 21 days, we slightly increased the carbohydrate intake and slightly decreased their fat intake. Beyond that, the portion of the type of fat that was consumed was very similar throughout. The types of food, as I mentioned, I'm emphasizing this because it was such a tightly controlled study. So when we're looking at microbiome and we're really trying to identify how does diet impact it, we weren't just looking at macronutrient ranges, we were looking at every ingredient in that particular diet. The primary results from this study focused on how dietary fat and specifically saturated fat interacted with metabolic health in the presence or in the absence of carbohydrates. And what we found is that when individuals were following the ketogenic diet or lower carbohydrate diets, even with increased fat levels and even with increased saturated fat levels, of the fat content was nearly 40% saturated fat, so particularly high, these individuals still improved on their metabolic health markers, and fairly uniformly, as carbohydrate levels increased, dietary fat decreased, remember, still eucaloric throughout the study, metabolic health seemed to reflect the increase in carbohydrate. So we didn't change calories throughout this. It was really that relationship between dietary fat and dietary carbohydrate. And when we looked a little bit further, we wanted to see how the microbiome was going to be reflected in this as well. We started out as a pilot study. So we took the entire samples that we had and really just focused on these first 2 time points, which were the individual's baseline diets, and then 6 weeks after we had been cooking for them in their well-formulated ketogenic diets. And if you're thinking about the timeline we discussed, within that 6 weeks, they're in that nutritional ketosis phase. So we started with the pilot study where we took 2 individuals. We tried to find 2 individuals of the study that were very similar in other factors, and again, following the same dietary approach. We collected stool samples and extracted 16S, specifically V1, V2 of the 16S region. We were able to sequence this particular lot through 454 technology. From these, what we found is that individuals had completely different baselines. So this is just 2 subjects, but this was our pilot data. They had completely different baseline data. Both subjects shifted with the changes in diet. However, there wasn't a uniform approach to how they shifted. In fact, one of them increased in Bacteroides, which is typically associated with a healthier microbiome, and one of them shifted in Firmicutes, which is typically associated with obesity. And so we weren't able to really identify many patterns, but we were excited to see that we are seeing shifts in the microbiome related to diet. So what we decided to do is extend this into a larger base study. And so wanted to extend this into the larger base study. So what we did is we took 8 of the subjects and we took 8 of the time points. So there were 6 feeding stages, 8 time points with that pre and post included as well. And so what we did is we pooled, it was an 8-pool gasket on the 454. We sequenced to try to identify any patterns. And what we found is very similar to our original pilot study, is at the phylum level that these individuals changed throughout the study. There were definitely dietary impacts. However, we weren't able to identify any specific patterns. We weren't able to identify any type of microbiome range that was reflected of the phenotypic changes that we were seeing. We weren't able to observe these deeper changes that we were looking for. We learned quite a bit through this study. In this study, we found that, again, the positives is that it's a highly controlled feeding study. Related to the microbiome, we know that diet impacts it in some way. We know that even slight differences are impacted. But we tried to remove that bias as much as possible and try to keep this controlled setting. And even in this setting, we still found that individuals wildly responded in terms of the microbiome patterns. It really was, on the research side, quite fascinating, but also a bit frustrating to not be able to identify any of the patterns. But we did have some limitations in this study as well. What we found is perhaps our sample size might have been too small to really start to dig into it deeper. Again, we took this as a pilot study, but we're still hoping to find some patterns that we could identify, but perhaps we need a larger sample size. Or the other aspect, which is what we're learning about microbiome-based research, is that perhaps this habitual diet means something longer than 3 weeks. And so since this was a secondary aim of the study, we were looking to see if these shifts were observed at the same speed as our phenotypic data. And we have a rich phenotypic database that we collected with this particular study, but 21 days might not be enough for us to observe these uniform changes. Or perhaps individuals are just that personalized that we may not see uniform changes in the microbiome. I think something that's really interesting is how this work compares to other areas. Again, there hasn't been much done in ketogenic diets, in microbiome, and metabolic health specifically. We see ketogenic diets in epilepsy research, and in fact, it's sometimes used as a way to manage uncontrolled epilepsy. So absolutely an impact that it has. But as it relates to metabolic health, often we see, even in the context of a ketogenic diet, categorized under this high-fat diet for the microbiome, it's typically talked about as a low-fiber diet. It's typically talked about one that may improve these other areas, but because of the food options that we're consuming, might not be representative of what we typically see with positive microbiome and gut health. We also tend to associate high-fat diets with these increased permeability, these worsening gut health markers and worsening overall health. So essentially we're categorizing this umbrella of high-fat diets without differentiating which high-fat diets are ones that we're properly prescribing and properly conducting to improve health, and which ones are high-fat diets that might just be poorly constructed. And instead, when we're looking at well-formulated ketogenic diets, again, going back to those initial slides, when done correctly, you are including non-starchy vegetables, you are including a variety of food and options, you are personalizing it to best fit the individual's metabolic health and overall goals. So when we're looking at it in this context, we start to consider what can future studies look like in this particular area. And so ketogenic diets in the microbiome, there's just so much to explore, there's so very much to learn. And there we start to learn and identify how does this properly and well-formulated ketogenic diet habitually consumed start to impact overall gut health changes. And it's interesting to know, is this going to be moving in a positive direction, or is this going to be moving in a direction that we're not sure how it'll impact the microbiome? And as we know with the microbiome, diet isn't the only thing that impacts it. The microbiome is sensitive to all external factors. And so how do these additional markers work in conjunction with diet to start to support positive gut health? So ultimately from this particular study, we just have much, much more to learn. And so we just covered a lot of topics in a lot of different systems of the body, but really the crux of what we're finding is we work in dietary prescription. We work with individuals that we're trying to learn more about the body or learn more about how individual aspects of the diet or how habitual diet starts to impact the system. And when we're talking specifically about well-formulated ketogenic diets, how does this unique metabolic state impact total physiology, which we're still discovering the extent of what that is, and how we're able to see it in a systems level. Specifically for our talk, we looked at gene expression levels where we can see parts of those keto-adapted phenotypes related to energy metabolism, and then it might also point to some of the more extensive areas. And then how can we use this information and also learn about it within gut health and the microbiome? And a lot of what the work demonstrates is just how much we still have left to learn. And so for these final clinical applications, we really wanted to better describe that relationship between metabolic health and microbial communities, and just know that it's such a close and synergistic relationship, and then how this adapts to the well-formulated ketogenic diet and metabolic health, and understanding that there's phenotypes that are observed in individuals that follow this dietary prescription or dietary lifestyle, and from there, what's the extent of it and how to best use it to help support the individual, whether that be a health or performance goal. So thank you so very much to you and to all the collaborators that have helped support this work, and excited to see questions.